Good afternoon. I'll be talking about prokaryotic transcription, specifically positive inducible transcription. So for positive, the protein acts as an activator, meaning that transcription is activated when the protein is bound to the DNA. And with inducible, it turns transcription on, so when the small effector molecule or just the effector is present, it turns transcription on. So also with positive inducible, the binding of the CAMP or the CAP complex enhances binding of RNA polymerase to functional levels, so it just speeds up transcription. So with positive, the protein binds, which is equal to up Tx. And the activator, the CAMP or the CAP, binds to the DNA and transcription increases. So with inducible, the small molecule binds as well, so CAMP binds to CAP and transcription increases as well. So if the small effector molecule is present, it binds to the activator and transcription occurs. But if it's absent, nothing can bind to the activator, so transcription cannot occur. So we have the regulatory protein binding site with an active activator protein, so it cannot bind to the protein binding site, meaning that nothing can happen. You have your promoter region, RNA polymerase can't bind to your promoter region, therefore transcription cannot happen. So when the inducer or effective molecule is added, you have your binding site, you have your inducer, so it's an active activator protein now. You have your promoter, your RNA polymerase can now bind to your promoter, and transcription can happen. As an example, you need two things to have a fire. You need matches and wood. The wood is what able to get caught on fire, but it cannot be turned on or started unless the match sparks the fire. Therefore, the wood is the activator and the match is the effector. Wood burning represents transcription. So you have your regulatory protein binding site, you have the inactive activator protein, which is the wood. It cannot bind to the protein binding site without its small effector molecule, so you have your protein, or you have your promoter, and RNA polymerase, it cannot bind to the promoter region, so transcription cannot happen. When the inducer is added, you have your structural genes and you have your regulatory protein binding site. When you add the match, the active activator, it becomes an active activator protein, so the wood has its small molecule on it. And promoter, RNA polymerase can now bind to the promoter, transcription is activated, and the fire is started. Here are my citations.